So if we look at the differences between this reactant and this product, the first difference you should notice is that there's a phosphate in the reactant and there's no phosphate in the product. The other thing to notice is that this left hand of the group and this right hand of the group is kind of very different. We don't have a carbonyl on the left or in the initial, but now we do on the product. And we had a double bond on here, but now we don't have it on the second part. And so to kind of explain this mechanism much more directly, I can show it here. So I will draw it. I apologize if my drawing is a little weird because I'm doing it in the computer. But if I were to draw this phosphoglycerate in like how we normally learn it in organic chemistry, it might look something like this. And I will just symbolize the phosphate group as a P. So this is our 2-PG, our 2-phosphoglycerate. And so the first step is that we get rid of, oh, sorry, not 2-phosphoglycerate. This is a phosphoenolpyruvate. So this is PEP. The first thing that happens is that the ADP transfer reaction happens first. And so the first thing is that ADP comes in and becomes ATP. And then as a result, this PEP will lose a phosphate. And so that will then turn into this. And then, if you remember organic chemistry, when an enol group is present, it almost always tautomerizes into a ketone. And so the next step is a tautomerization. And so this enol will become a carbonyl. And now we have this product. And this happens to be the pyruvate that we have as our final product. And so this last step right here, even though we call it a kinase step, it's actually a kinase step followed by a tautomerization step. So I hope that clarifies why it looks this way. Okay, so now we're at the end of glycolysis. I've talked about all of the mechanisms and like the enzyme names. So I hope you guys can look back to this video if you're not sure about that step. But now let's look into the energy part of this pathway. And to look at that, let's look at the kind of pathway as a whole again. We have the first part of glycolysis, which we spent 2 ATP. So we call that the investment phase, so minus 2 ATP. And then the next phase, we generate ATP as a result as well. And in this pathway, we generate 2 ATP, but remember, because the sugar is split into 2, this reaction happens twice. And so as a result, the amount of ATP we get at the end is 4 ATP. And so if you invested 2 ATP and then you harvested 4 ATP, your net ATP is 2. And then you also harvest 2 NADHs. And so as a result, you have 2 NADH harvested. And you guys will know, already know, the NADH will be also a source of energy. Okay. Before I move on to regulation, do you guys have any questions for those of you who are in the recitation? Okay. So let's move on to like the regulation now. So if we look at a delta G graph of glycolysis, so a couple things to observe is one, the delta G is overall negative, which makes sense. This reaction wouldn't happen for us if this was not spontaneous. But the other thing is to note is that there are a couple steps in glycolysis that are very delta G negative. And so in particular, we have this first part right here, hexokinase. Then we have PFK, that's also very negative. There's also PK right here. And so one thing to note, why are these steps very delta G negative? And the first thing is that the hexokinase step is very negative because of this ATP investment phase. Then for phosphorufractokinase, the same thing. There's an ATP invested. As a result, the delta G is very negative. So it's a very irreversible steps. And then for PK, this is where the ATP is harvested. And this step is also considered irreversible. So to kind of summarize, an irreversible step is a step where the delta G is very negative. And also, because these steps are so-called irreversible, they also are known to be points of control. So if you ever want to control how much this pathway runs, you need to look into these steps because they're the ones that really affect the pathway as a whole. And so kind of explaining that in an energy diagram, to talk about regulation, so the first general rule that we want to say is the products of a pathway almost always inhibit making more of that. And so if we look at the glycolysis pathway we have here, I don't know the exact listing of inhibition, but generally we should expect that pyruvate should in some way maybe inhibit this enzyme, or maybe this enzyme right here, or this product right here, probably likely inhibits the enzyme that makes it. And so you'll almost notice in biochemistry from now on that almost all pathways that we talk about in the future, if they ask you what's an inhibitor of that, it's almost always the product. And so this is known as feedback inhibition, where the product of your reaction actually inhibits the production of more of that. So that's one general concept in inhibition in general. The other thing to note is that when you actually inhibit or regulate a mechanism of an enzyme, there's a couple things you can change about that enzyme. You can affect it biochemically, or you can affect it almost genetically. So the first thing is you can change the Vmax or the Km of your enzyme. So if you guys remember your inhibitors from YAPIs you have allosteric inhibitors, you have your competitive, uncompetitive, and all of that stuff. You can change the Vmax or Km and alter how fast this mechanism occurs. The next thing is to produce different forms of the enzyme. So you can have different isoforms that you can produce. So if you produce more of a slow isoform than a fast isoform, then you can change the rate of the enzyme production. What I mean by the slow and the fast over here is that inhibition with an inhibitor, the response time is relatively fast. The moment the inhibitor binds, you see the inhibition. But if you have if you want to make different forms of the enzyme, this implies that you have to transcribe the gene, then you have to translate the gene. And so as a result, this process takes longer. And then the most easy way, if let's say you want to just downregulate a pathway, is you can degrade the enzyme, or you can phosphorylate it. And if phosphorylation inhibits the enzyme, that works perfectly in our case. And so to kind of give you the examples earlier, connecting this with the graph, the most obvious ways to regulate glycolysis is through the three irreversible steps we talked about. The hexokinase, the glucose 6-phosphate inhibits hexokinase perfectly. The phosphoenol pyruvate, which is a product of phosphofructokinase, it directly inhibits phosphofructokinase. ATP also inhibits phosphofructokinase. And why this makes sense is that if you're in an energy high state, if your cell has a lot of energy, you don't need to do glycolysis. And so it makes sense that the ATP will inhibit the phosphofructokinase. And the citrate we'll see in the future is a kind of downstream product of if we do the citric acid cycle. And so inhibiting PFK through citrate also makes sense. These products, in the meanwhile, on the right, AMP, fructose 2,3 bisphosphate, and fructose 2,6 bisphosphate, they activate PFK. And so why does this make sense? When you have high AMP, that tells you that the cell has low energy. And so because the cell has low energy, that means you want to do glycolysis more. And so as a result, it makes sense that AMP increases PFK. The F23BP and F26BP, those are intermediates of glycolysis. And if you guys remember, so actually F23BP is a kind of variant of this F16BP. And so generally, these will show later, but they're not directly part of the pathway. But these are markers of high energy, or sorry, low energy. And when they're markers of low energy, that basically encourages glycolysis to occur. And then for pyruvate kinase, PK, it's inhibited by alanine and it's encouraged by fructose bisphosphate. So this first part right here, it's encouraged by fructose bisphosphate. Does it make sense in this case? Because the fructose 1,6 bisphosphate is a reactant, or in this case, it's kind of upstream of glycolysis. And so it makes sense that the high amount of this means that we need to run the pathway. So think of it like the Shotlius principle. If you have a reactant to product reaction, if you have a lot of reactants, it makes sense that the pathway is shifted to the right. And so that's why it makes sense that fructose BP activates pyruvate kinase. For the alanine, what you might not notice is that pyruvate is actually a precursor to alanine. So we'll learn later on in the future that through a series of steps, pyruvate gets turned into alanine. And so if you have a lot of alanine, that tells you that you have a lot of pyruvate. And if you have a lot of pyruvate, you don't need to do glycolysis. And so as a result, this pathway is not needed. Hence, we can inhibit pyruvate kinase. I hope that makes sense. In the case of the exam, how you might get asked about this is more of which step of glycolysis should you regulate, and also what kinds of things should you are expected to regulate. And so be prepared to be asked which steps are regulated, but also what kinds of things would regulate it. What would happen if I add a bunch of alanine to my cell? Would glycolysis be increased or decreased? That's very important.